All right, well, our next speaker is Dr. Jason Karnes. Dr. Karnes is an assistant professor at the University of Arizona College of Pharmacy in the Department of Pharmacy Practice and Science. He also has appointments in the Sarver Heart Center as well as the Division of Pharmacogenomics. Interestingly, Dr. Karnes started out his educational trajectory studying ancient Greek at the College of William and Mary, and then went on to obtain his PharmD and PhD at the University of Florida. After that, he did a postdoctoral fellowship at Vanderbilt. So he's had training along the way in two of the sort of the flagship institutions doing a lot of implementation work in the area of pharmacogenomics. He's really made a name for himself in advancing the ways in which we think about issues of racial and ethnic diversity, as well as genetic ancestry in looking at drug response, as well as predisposition for serious adverse events from a variety of different medications. His research work is supported by foundation grants from the American Heart Association as well as the American College of Clinical Pharmacy, and it's a real pleasure to have him here today. Join me in giving a warm welcome to Dr. Jason Karnes.
Thank you, Jeff, for that very kind introduction. So my main goal today is to have everybody that's here walk away with an idea of the important and critical role that race and ethnicity is going to play in pharmacogenomics. But before I do that, first I want to tell you a little bit about myself. I am 98% European, or 99.8% European with less than 0.1% South Asian ancestry. I am 37.8% Scandinavian, and I would like to specifically point out that I have fewer Neanderthal variants than 70% of 23andMe customers. So really, I'm going to start the talk by talking about a concept called population stratification. And so this is one of the sources of bias that we have in terms of race and ethnicity in all genomic studies, not just pharmacogenomic studies. So take this figure where we have two populations. One population has an overabundance of cases, right? So this happens very commonly. For instance, type 2 diabetes in Hispanics. It's more common in Hispanics than it is in several other race groups. So when you do your association study and there's an overabundance of cases within a population, you may well be tagging variants that are common to the population as opposed to the disease. Right, so you do your GWAS with Hispanics in type 2 diabetes cases and controls, and you're identifying variants that are specific to Hispanics, not variants that are specific to diabetes. And this population stratification, it is one of the reasons that we have a bias in genomic studies. Genomic researchers typically want very homogeneous populations, typically European, that are simple to study and the results are simple to interpret once you have them. And this is one of the major reasons why most of the literature in genomics is in patients of European ancestry. You look at these numbers, these are staggering numbers. So from 2009, 96% of all individuals in any genomic study were white. And that has improved slightly. So you see the 2016 numbers. It has improved slightly. That's due primarily to an increase in the number of studies of Asian individuals. And you can see the breakdown on the right side here that is primarily Asian, but studies in individuals of African ancestry, of Hispanic ethnicity, they lag far behind. This is a study that was done in 2018, and you see very similar data. Just an aside, I don't really like pie charts. Just give me the numbers, the proportions, but you're going to see a lot of pie charts in this presentation. In terms of the individuals, we have similar numbers as the previous slides. So 78% versus that 81% I showed before are white. But in terms of the associations, in terms of the genomic associations that we identify, and this is data from the GWAS catalog, there is a disproportionate number of associations that we find in African individuals, in mixed race individuals, in Hispanic or Latin American individuals, suggesting that there's the potential for more discovery within these race groups. And so despite the fact that we have such a large proportion of our genomic research being conducted in Europeans, there really is the possibility of this greater juice from the squeeze, for instance, in terms of genomic associations. But there's an issue here because precision medicine, there's the possibility of there being a disparity. So there are already disparities in terms of healthcare. There's the possibility that precision medicine is going to widen these disparities based on access, based on the amount of evidence that we have in Europeans versus other race groups. Now, this can be due to the fact that we're not really characterizing variants very well in these more minority, in these diverse race groups. We are not developing models like algorithms. I'll point out an example with warfarin, where our algorithms just simply aren't as good or weren't as good in non-European individuals. There's also the question of cost effectiveness and clinical effectiveness. And do we need to study, we mentioned RCTs earlier today. Do we need a separate RCT for every drug-gene pair and a separate RCT for every drug-gene pair for every race and ethnic category that we study? Now, that's not necessarily true, but it will be very important to have representative studies that are inclusive of all the race groups that we want ultimate clinical implementation to affect. The bias in these genomic studies, it's particularly concerning because these minority groups have a potentially greater impact in terms of pharmacogenomics. There's some evidence here, so warfarin dose variability is increased in many diverse populations, and in addition, non-whites also have a higher risk of warfarin-related cranial hemorrhage. So not only are adverse effects increased, but variability is increased in some of these minority populations. Yet, with warfarin, this is the clinical trial data. So this is an article from last year, from 2017, but you see a very similar picture. 80% of the individuals represented in the clinical trials published for warfarin pharmacogenetics were European. African-American individuals follow closely behind, as well as Asian individuals, but you see a very similar paradigm of underrepresentation of these diverse race and ethnic groups and a potential widening of the health disparity that we have in genomic medicine. Now, I mentioned this can be due to changes in frequency. Certainly this is one of the major possibilities. So in this table, I have the minor allele frequencies in different race groups of very common warfarin pharmacogenetic variants. And you can see I've highlighted some of the CYP2C9 variants, so star 5, star 6, star 8, and star 11, all of which are part of the CPIC guideline, the most recent guideline that came out. And you can see they're essentially absent in these other populations, yet they exist within the African population. You can look down at the second from the bottom, the minus 1639 G to A substitution, and look at the vast differences in terms of frequency of that variant between race groups. From a 5% sensitive variant in African race groups to an 88% in those Asian race groups. And this data is some of what clinicians have known for decades, is that different race groups respond differently to warfarin. Africans typically have higher dose requirements, weekly dose requirements, and Europeans and Asians typically have lower ones. And this partially explains that phenomenon. The studies that have looked at pharmacogenomic dosing algorithms in these diverse populations have more or less consistently found that the algorithms developed within European populations, so this represented here on the right as the International Warfarin Pharmacogenomics Consortium algorithm, when race-specific algorithms are developed, and there's a number of populations represented here, African Americans, Hispanics, and Asians, you can typically see an improvement in terms of inclusion of genetic variants that weren't included in the IWPC algorithm, as well as clinical variables that were used for these study-specific populations as opposed to IWPC. So this data really suggests that European-based algorithms are not necessarily generalizable to other ethnic groups, to African Americans, to Asians, to Hispanic populations. And that's consistent with a finding that's from 2013. This was a genome-wide association study exclusively in African American patients. And so a entirely new SNP, well, a new association with warfarin pharmacogenomic dosing was found. And this was subsequently incorporated into the CPIC guidelines because it had a 21% improvement in terms of the prediction of warfarin stable weekly dose in African American patients. But this is a variant that had not been shown to be associated within the European populations. So already we're finding variants that have a higher frequency within African American populations that would not have been included originally in the IWPC algorithms. And again, this is reflected in the CPIC guidelines. So my purpose is not to go over this entire figure, but you can see that one of the first steps is to divide individuals by race. So this is self-identified West African ancestry, right? And there are issues certainly with self-reported race, and we'll go into some of those. But one of the bottom lines is that race is complicating this pharmacogenomic picture. There are variants that we now know affect warfarin dose variability in some race groups, but not others, or at least are of a higher frequency in some race groups besides Europeans. You can see that the GWAS SNP, as well as those CYP2C9, star 5, star 6, star 8, or star 11 alleles are included on the African side of the algorithm, but they're not included on the non-African side, or at least included in an optional way in the non-African side. By the way, the last 3 slides I presented were the first or corresponding author was Julie Johnson on each of those last slides. So I'm definitely riding her coattails with this presentation. So I've mentioned differences in frequencies between race groups. Certainly there can be differential effects, although that hasn't been seen, that hasn't really played out in the literature quite as much. So we have a couple of options in terms of an approach. We're thinking about implementation now. So we can do this cosmopolitan approach, right? We try to character all the variation for each race group, and we try to implement it such that all race groups benefit. There's obvious issues with this approach, one of which is that currently our evidence is lacking in terms of diverse race groups, in terms of inclusion of African Americans, of Hispanics, and of all the genetic diversity across the globe. Remember, there's still 80% of the people in our genomic studies are of European ancestry. And that might not be the most cost-efficient approach because, as we said, some variants exist within diverse populations that are associated with pharmacogenomic variants or pharmacogenomic outcomes, and they may not need to be tested in individuals that don't have that ancestry. But therein lies another problem in that how do we assess ancestry? Is somebody's reported ancestry, what they report, whether they say they're black or white or have Hispanic ethnicity, how much information does that contain on a genomic level, especially considering how much admixture there is in the US population and in the world population as it continues to increase. And so that really ties into some of the issues with the self-reported race. I mean, these are Office of Management and Budget standards that are commonly used, but they may not capture that increasingly admixed population. Again, we have a lack of evidence in non-Europeans, but these definitions are convenient, and they often characterize much more than just genomic admixture. Individuals who report certain ethnicities or certain race categories, that comes with a whole a whole lot of more information, including socioeconomic status potentially. It includes potentially diet habits. And then we can get more specific. We can get into national origin, or we can use a more objective measure. We can look at the entire genome or certain ancestry informative markers across the genome and then create a continuum. Each individual has a percent European, percent African, percent Asian amount of ancestry, and they can either be grouped based on these objective markers or put on a continuum where an increasing amount of European ancestry suggests that these certain variants become more and more important. And just an aside, just think about how much genomic diversity there is within the Latin or Hispanic population. Those terms are not perfect. There's a Venn diagram, and they don't encompass all individuals. But there is a staggering amount of diversity in terms of the amount of African ancestry, the amount of Asian ancestry, and Native American ancestry present in these groups. And so simply checking a box that says yes or no for Hispanic ethnicity is not going to tell you what you need to know in terms of these pharmacogenomic algorithms. I mean, it's valuable information, but there is such a diverse group of Hispanics in the world today that becomes exceedingly difficult to implement in terms of an algorithm. So switching gears here a little bit, one of my favorite topics is HLA. So human leukocyte antigen, we've mentioned it several times up to this point. But the reason I bring it up is because HLA has a tendency to be very, very race specific. So because we're talking about a whole gene, all of the variations within that gene, so really a haplotype for a whole gene, you get specific 4-digit HLA alleles that are unique to certain populations, right? And we're still finding new HLA alleles out there. The last IMGT, I think, had around 12,000 HLA alleles, class 1 and class 2. But the staggering amount of diversity means that we haven't characterized all the alleles yet. Yet there was this FDA-issued warning for Carbamazepine and B1502. So what this essentially said was there's an increased risk of Stevens-Johnson's toxic epidermal necrolysis, which is very severe, very bad, 30% mortality, 30 to 50%, I think, last time I checked. And, but it's found almost exclusively, this allele, in patients with Asian ancestry. So, okay, that's helpful. So maybe we only need to test in patients with Asian ancestry, right? But what does that mean exactly? Anybody from Asia, anybody who self-reports as having Asian ancestry, and does the admixture within the US make that readily implementable? Can we really use that data to do a race-specific algorithm based on this HLA? This is a map of Asia. So, and there's X's in all of the other. So this is a published article. I don't know that they really needed these figures, but you know, they could have just said it is restricted to Asia. But so this is the area covered by the FDA's pharmacogenetic warning. Yet this is the area for the evidence that was used to generate it. So there is. Now, I'm not saying that the warning is incorrect, because the Asian ancestry, it is, 1502 is largely restricted to Asian ancestry, but we just have to, we have to realize where this evidence is coming from and make sure that we're implementing it in a way that we're covering all of the individuals that are potentially affected by these guidances. And there are all kinds of these associations. Not all of them are clinically implementable because some are so rare, or some are, don't really have a good predictive ability in terms of predicting whether you're going to get SJS. Abacavir is a notable exception. Supported by a randomized control trial, it is now standard of practice to use HLA-B 5701 testing for abacavir to prevent hypersensitivity syndrome. But you can see the differences in global frequency of these alleles. So 5701 is in red. You can see how that's, in some regions in Asia, it's relatively, it's very low frequency, yet in the US, it's fairly high frequency in European populations. And is this perhaps one of the reasons that abacavir was so readily uptaken, is because the evidence was so strong and it was done in European populations? That's a possibility that has yet to be explored, but many of these drugs were developed within the US, where there's a relatively low Asian population, and then they were subsequently used in Asia, where these alleles were more common. And so one of the reasons that these drugs got through an FDA approval without so much knowledge about Stevens-Johnson syndrome, toxic epidermal necrolysis, is because we were using them in a fairly European population, and then when we transported them to Asia, where there's a much higher HLA frequency, we started seeing more and more SJS and TEN. And this has prompted an implementation within the governments of Singapore and Taiwan. So for instance, Singapore, they have implemented HLA-B 1502 testing, and they reduced the number of cases from 18 per year to 1 case in the 4 years since implementation. So an extremely successful, and again, these are low numbers, but this is extremely severe when it does happen. And this is a state-mandated implementation. In Taiwan, we have similar success stories, but I just want to mention there is a different allele that's been associated with carbamazepine-induced SJS in Europeans and other Asian populations, and that's A3101. So now we're introducing a new complexity. Are there different, perhaps, variants that are specific to different race groups that can influence whether or not we're able to implement in these groups? Obviously, cost effectiveness is an issue. In places like Taiwan and Singapore, where these variants are very common, it's going to be more cost effective to use this HLA screening. But the importance still exists to incorporate multi-ethnic populations. We can't just assume that an algorithm that we develop within Europeans is going to work just fine in Black individuals and Asian individuals. So we have to generate the evidence. We have to look for new variants. We have to discover new variants within those ethnic populations and incorporate them into our algorithms. And again, the old approach of defining race and ethnicity based on 7 questions in the OMB is not likely to be as informative moving forward. And so an objective measure of race and ethnicity is probably going to be necessary. There are a number of new approaches. We can now combine data. We can avoid population stratification even though we're combining all of the race groups and looking at that combined data to discover new associations, new variants that affect pharmacogenomic outcomes. Now, these are primarily meta-analyses techniques, which are really nothing new, but at least the public is starting to invest in this, and we're starting to really try and close the gap in terms of the health disparities with the genomic associations. And again, this approach of the multi-ethnic association analysis has been applied successfully and generated novel signals for a number of different diseases to this point. So at U of A, we are an all of us site. The reason this is important is because of what I mentioned about closing those gaps, right? So all of us has the goal of including 50% of underrepresented and biomedical research individuals. So 50% of all of the 1 million patients that get recruited should be from an underrepresented population, whether that be racial or sexual orientation, or there's a number of different categories that you can fall into to be underrepresented. And at U of A, we've actually recruited the most of any of the current sites. And 90% of the people that we've recruited from the U of A are underrepresented in biomedical research. Now, we've only recruited 7,300, and I think the mark is somewhere like 150,000, which is going to take a while. So, of course, you have recruiting centers here, but if you'd like to come to Arizona and have us take credit for your enrollment, please do. That would be no problem with us. So in terms of concluding remarks, again, I want to underscore the strong bias that we've seen in the literature, and that we're improving, we're going in the right direction. There's a number of NIH initiatives that are pushing us in the right direction, but we continue to lag behind in terms of the available evidence in these ethnic minorities. Again, extrapolating the results that we find in Europeans to other ethnic groups is not necessarily going to be a valid approach. And we have many approaches for discovery now where we can incorporate multiple populations and still avoid population stratification. And again, including all of the diversity that we have within the United States and the world, it's going to be essential to ensure the benefits of pharmacogenomics and of precision medicine are universal, and that precision medicine doesn't end up widening the health disparities gap that currently exists. And with that, I will take any questions. Thank you for your attention. Yes.
Thank you. Yeah, I had a question. So to my understanding, the newer build of the reference genome accounts for a lot of ethnic variation with alternate scaffolds. I just wondering if you could speak to how well commercially available genetic testing panels have kept up with this as new markers are added.
Well, I think, so for instance, I was looking over the 1-Ohm report, and the GWAS variant, the 127, I forget what the number sequence was included, right? So clearly updating is happening for the more responsible purveyors of direct-to-consumer testing. The build issues are definitely getting better. I mean, where we typically had a European genomic build, essentially, the new scaffolds, as you say, have improved our ability to interrogate the reference genome when we're looking at multi-ethnic populations. And the reference groups for imputation or HLA imputation have gotten much better, but there's still a relative lack versus the Europeans. I don't know that will ever change completely, but certainly as the evidence comes out, the genetic platform should be updated to be inclusive for all the race groups. Does that?
Thank you.
Yeah, sure.
Hi, Jill Larson. I was just going to address a similar thing, whether you go retrospective back with maybe studies or manufacturers of different medications and looking at alleles or some of the genetic things where like perhaps medications have been taken off the market because of side effects, when for a certain group they might actually be really useful?
Yeah, no, that's a great suggestion. I mean, obviously there are a lot of issues with going back to some of the landmark clinical trials for drug approvals. Many of those studies may not have genetic samples available, although the FDA is asking drug companies to keep samples. So there's an interesting example for lemiracoxib, which is a COX-2 inhibitor, was approved in Europe. And one of the reasons it failed was because of a drug-induced liver injury. Well, as it turns out, there's an HLA marker for that drug-induced liver injury. Now, the drug company never actually picked that up. They never used a companion diagnostic genetic test for a COX-2 inhibitor, probably because there are already other COX-2 inhibitors out there and they didn't really see a market for it. But there is that potential for pharmacogenomics to come in and save a drug that is getting thrown off the market because of an adverse event or not being approved because of an adverse event. And similarly, if you look at it as you suggest in multi-ethnic populations, look at Beidel, classic example, which is approved in a specific race group. Why can't we do this on a more objective, race-based manner?
Let's do one more quick question from Dr. Van Ness?
Yeah, given the cost continuum of different approaches in genotyping, that is, having separate panels that will be addressed to different ethnic populations, versus doing a whole genome or a whole exome, true or false, if we do a whole exome or a whole genome on everybody, this goes away. The problem goes away.
Well, I mean, I think you're right. I think it does. So true with an asterisk, maybe? I mean, it sounds very simple, right? But obviously, there are a number of ethical and bioinformatic challenges to making a whole genome available at birth or at consent for an individual. But that's the Collins, esque dream, right? Is that eventually we get sequenced at birth or at some point in our lives, and this data is there to be extracted whenever we need it, whenever a drug is ordered. And of course, that data can give us very, very fine-tuned information about race and ethnicity. I mean, it doesn't capture the socioeconomic status and the diet implications of those categories, but we can certainly define race based on whole genome sequence data. Yeah.
All right, Dr. Jacobson is going to offer some words before lunch, but before she does, I'd like you to join me in one more round of applause for Dr. Karnes and Dr. Beck.